Welcome back, everyone, from lunch. And it's great to have everyone here for our afternoon session on latency and reservoirs. And we're going to be started off by Pam Skinner, who's here from the University of Minnesota, and she'll be talking about CAR CXCR5 T-cell immunotherapy in SIV-infected, ART-suppressed rhesus macaques.
Good afternoon. Don't you just love this meeting? It's such a good meeting. It's my favorite meeting. I'm so glad I'm here. I'll try not to spoil it. So today I'm going to tell you, as mentioned, that I'm going to tell you about our CAR CXCR5 T-cell immunotherapy project that we're doing in SIV-infected macaques. And to begin, I want to start with some of the rationale for which this study is based. And so we know from many studies that during HIV as well as SIV infection, the virus becomes concentrated within secondary lymphoid tissues and CD4 T cells. And we know from work from Liz Connick from over a decade ago that within lymphoid tissues, virus replication becomes concentrated within the B cell follicle. And Liz, a while ago, formulated the hypothesis that perhaps a mechanism mediating virus replication concentrating within the B cell follicle is the inability of virus-specific CD8 T cells to get into follicles and clear those replicating cells. So she formed a collaboration with me because I had developed a way using in situ tetramer staining to localize virus-specific CD8 T cells in tissues. And we collaborated for a decade looking at these questions, and we looked in both lymph nodes from human subjects as well as in the SIV rhesus macaque model system. And what we found is that virus-specific CD8 T cells were indeed most concentrated out in the T cell zone, and in many instances we would find follicles, as shown here in green, for which no virus-specific CD8 T cells were present. So to orientate you on this slide here, there's a lymph node here. It's stained with CD20 antibodies to label B cells green and to delineate B cell follicles, which are shown here. The virus-specific CD8 T cells are labeled red, and they're indicated with arrows. And so you can see in this first lymph node, all the virus-specific CD8 T cells are outside the follicle. In this one here, which is typical of what we'd often see, is you see some within the follicle, and they're typically along the edge near the T cell border. But then we can also find examples where there's similar levels within an animal or a human within a node. You can find a follicle generally that has similar levels inside and out. So they're not entirely excluded. They're just in lower levels. But nonetheless, when you think about in vivo effector to target ratios, inside the follicle, there's a 42-fold lower effector to target ratio than outside of the follicle. So these data indicate that the B cell follicle is somewhat of an immune privilege site where low levels of virus-specific CD8 T cells permit ongoing viral replication. In the study, we also found that levels of virus-specific CD8 T cells within lymphoid compartments inversely correlated with levels of viral-producing cells. So this indicated that, one, the cells were functional, but also that numbers matter. In other studies we've done, one with Ashley Hawes over a decade ago, we found that in vivo effector to target ratios were positively associated with reductions in viral load in the SIV macaque model system. And more recently, we've shown in a couple of different studies that levels of virus-specific CD8 T cells in follicles and outside of follicles inversely correlate with viral loads. So numbers matter. And these studies led us to hypothesize that perhaps increasing levels of virus-specific CD8 T cells inside follicles may functionally cure HIV. And to begin to test this hypothesis, we are transducing virus-specific CD8 T cells with the B cell homing molecule CXCR5. And so in order to make the CXCR5 cells virus-specific, we've been collaborating with Dr. Ed Berger from the NIH, who's here at the meeting. And for those of you who don't know, Ed's made a chimeric antigen receptor that's bispecific. It has 2 HIV binding regions. And he's also produced a rhesus macaque version of this molecule that will bind SIV. And so we've formulated a construct in a gamma retrovirus that has EDS-CAR and also co-expresses CXCR5. And again, so the CAR, when the CAR is expressed, that provides that T cell with the ability to recognize and kill virus-infected cells. And when the CXCR5 molecule is expressed on the cells, that allows the cells, directs those cells to home to the B cell follicle. So what we've done now is we've taken our construct and we've transduced primary PBMCs from rhesus macaques. Mary Pampush in the lab spent some time with help from people like Chris Peterson and others to develop a method to transduce and expand these cells in 9 days and maintain a central memory phenotype. Then she's evaluated these cell products with flow cytometry and found that they're highly viable. She can get very high co-expression of both the CAR as well as the CXCR5, and she's maintaining the majority of the cells with a central memory phenotype or a less differentiated phenotype. We've also gone on to examine the function of these cells in vitro and ex vivo, and using a standard transwell migration assay, we found that the CAR and CXCR5 transduced cells specifically migrate to the ligand CXCL13. And we've also developed this novel ex vivo tissue migration or follicular migration assay in which we've taken transduced cells, and we label them with a live cell stained CTV, and then we pipette those on top of a fresh slab of lymph node and incubate that for 6 hours, then fix the tissue and stain it with antibodies to CD20 and CD3, a T cell marker and B cell marker, and then quantify the cells inside and outside of follicles. And Guanto in the lab did this, and she, here the green again are the follicles, and the arrows are indicating the CTV labeled cells, which are pseudocolored yellow here. And what she found when she quantified is that the cells that were transduced with CAR alone preferentially homed to the extrafollicular areas, similar to what you would see a normal cell do, whereas the CAR and CXCR5 transduced cells preferentially homed to the B cell follicles. Then Ed Berger and his group, he looked at the functionality of these cells and determined in a vitro suppression assay that the CAR and CXCR5 transduced cells effectively suppressed viral replication in vitro. So with that, we then went on to start testing these cells as an immunotherapy in the SIV infected rhesus macaque model. And we want to see is this therapy safe? And do the cells migrate to follicles? Do they induce the suppression of viral replication, reduce viral loads, and importantly, do they lead to a functional cure? So with our first study animal, we had a long-term non-progressor who was starting to progress, and we infused a relatively low dose of cells, about 350 million per kilogram, into this animal, and then 2 days later sacrificed the animal and examined where the CTV positive cells were located. And we found that we were in the spleen, we could readily detect cells in the follicle as well as in the extrafollicular area. We saw fewer cells in the lymph nodes, but we could readily detect them in the follicle as well as outside. We were happy to not see cells accumulating in the lung. That's been an issue with some folks, some studies in the field. And we also did not detect any in the bone marrow, liver, or brain, but did detect a few in the gut. We also saw something kind of interesting. Now this one, the colors have changed. Here, the CTV labeled cells are yellow, and the red staining is CXCR5. We saw evidence of proliferation of the CAR T cells in the T cell zones. So here's a cluster of cells replicating, and you can see them here. And we know that when the CTV when cells divide, the CTV signal becomes half as bright, so we can see very bright cells and half as bright cells as well as doublets. Not to mention this cluster of cells together here that all tell us that these cells are dividing. So that was quite interesting. Then with that, we went on and we did a pilot study with 3 treated animals and 3 untreated control animals. And for this study, we infected animals with SIV MAC251. They were ART suppressed. We collected cells during the chronic stage of infection and transduced and expanded them, then infused them and released the animals from ART the time we infused the cells, and then studied the animals for 2 months after infusion. And for this now, we've increased the dose from our first one, which was about 0.4 times 10 to the 8th cells per kilogram, and now our goal was 1 times 10 to the 8th cells per kilogram, and we got close with these animals. And so, again, we took biopsies 2 to 3 days post-infusion, and again, looked for those CTV-labeled cells. And here you can see many yellow cells here inside this follicle, so we're able to readily detect them in the follicles. And the increased dose led to more cells in the lymphoid tissues, which is great. When we looked at the viral loads, here's the 3 animals, and to orientate you, the blue is the treated animals, and the red are the matched controls that were done. In the first animal, we saw immediately, 10 minutes after infusion, a high viral load. It was reduced a little bit, and then it was lost. With the second animal, we had the sense to do viral loads right before infusion and 10 minutes after. And it became abundantly clear that with our cells that we had produced from chronically infected animals, when we infused them, we were also infusing virus. But nonetheless, in this animal, you can see that at the next time point, 2 days later, that virus was undetectable, remained undetectable for a while, and then the animal remained controlling better than its matched control. The third animal, again, a spike of virus was immediately suppressed, and then we saw a little bit of control here that was lost afterwards. But if we look on the positive side, one of the 3 animals looks fantastic and looks much better at controlling virus than the controls. And when we look at the median values for the study as a group, we can see what looks like a little bit of control early on. Next, we had funds for 3 more animals, and we decided to test purely proof of concept here. And so we collected cells prior to infection, so we wouldn't have any problem with infusing virus. And then we performed the same experiment. The animals were ART suppressed. We infused the cells, released them from ART, and then monitored them. And here now we also increased our dose again for these animals. We got up as high in one of the animals to 2 times 10 to the 8th cells per kilogram, which is, and there's a representative figure from this animal here. So now you might see with the increased dose, again, we've got a much higher level of these transduced cells within the lymphoid tissues and follicles 2 days post-infusion. And so here, what's also interesting from this one image is you can see this cluster of replicating cells here near the follicle in the T cell zone. And you might not be able to see it, but I can tell you that there's a whole bunch of cells over here that are way less bright than these cells here. So this is in the follicular area, and this is in the T cell area next to the follicle. So what these data are indicating is that these cells are very early after infusion, they're replicating in the T cell zone and then entering the follicles. Now with this study, the viral loads a little more promising. The first animal, so here now the treated animals are in blue, and the control animals, these are the same set of controls I showed you in the previous study. We're recycling them for this study. And you can see that the first animal had what looked like a little bit of control early on that was lost. And the second 2 treated animals had a much lower peak viral load and then went on to control. And if we look at the median for the study groups, we can see a clear difference between the treated and the control animals. So we're psyched about this. And if we look at 1 month post-infusion, we can see a substantial 2-log reduction in viral loads for the treated animals compared to those controls. Then we also, as I mentioned, we started out with a relatively low dose, and then we kept increasing the doses, and then we got up to 2 times 10 to the 8th per kilogram. And so we did a dose response curve, and we were really delighted to see that increased levels of virus-specific CD8 T cells were associated with significant reductions in viral load. So to summarize, we've tested our CD4 MBL CAR, CXCR5 T-cell immunotherapy and found it to be safe. And in vivo, we found that these cells proliferated in T-cell zones. They accumulated within lymphoid follicles. We saw a lower viral load overall in the treated versus the control animals. And we saw that increased dose of cells was associated with significant decreases in viral load. So this therapy seems to be safe and fairly effective. And to summarize for the community, our main question is, can we suppress HIV with a patient's own cells? And the progress we've made so far is we've grown killer T cells against the virus, or CAR T cells, that migrate to lymphoid follicles and suppress viral loads. And what does this mean? Well, it means that targeting killer T cells to B cell follicles could result in the ability to keep viral loads suppressed and undetectable without any medications. With that, I'll acknowledge the folks and thank the folks that contributed to this work. In my group, Mary Pam is here. She's really done most of the work on this project with the help from Yamari, who is a right-hand person who helps her get going. We've also got a new postdoc in the lab, Hadia Abdullah, who will be presenting after me and showing you her new results from this study, her RNA-scope studies. There was also several other members of the lab who have contributed over the last few years, as well as folks from the Believe Collaboratory. As I mentioned, this work was all initiated with Liz Connick. The folks, Ava Rakaz and particularly Nancy Schultz-Darkin at the Wisconsin National Primate Research Center have managed all of our primate studies. And also Ed Berger, as I mentioned, we've been collaborating with him for years in his car. Thank you very much.
Hi there. Maybe you or Ed have done this already. What about just the construct of the CAR alone? I assume that doesn't go to the follicle, but has anyone looked at that? And then whether or not there's an effect on viremia if it was just a CAR alone construct? Thanks.
So we only have done the studies shown. While we had CAR alone in, we just don't have the funds to do that. But I understand perhaps somebody here is about to do that study now. So we don't know the answer yet. But I would predict that it wouldn't work as well. Because you're not going to be targeting the virus, which is, the virus is concentrating in the follicle, and if you don't get there, you're missing the majority of the virus replication.
Anything else? One more question, Jim.
I'm sorry. Maybe I missed this, but how long after infusion could you find the CAR T cells?
Hadi is going to share that with you in the next talk.
All right, I'll wait.
We'll take that. Thank you very much.